So moving now to the first keynote talk, Chris Dowson is just about to share his slides. I can see those. Thank you, Chris. So great to have you here today, Chris. And Chris is Professor of Microbiology at the University of Warwick, where he runs the Warwick Antimicrobial Interdisciplinary Centre. He's also trustee and chair of the scientific committee of Antruc UK, where he's doing work to help support antibiotic discovery research across the UK and wider. Really excited to hear Chris talk today about new approaches required to address the failed and failing R&D landscape in antibiotic discovery, which is something that's clearly key to all of us that are working in antibiotic discovery and development. So over to you, Chris. Thank you.
Lovely. Thank you, Matt. Morning all. Lovely to be here and great to be part of a vibrant and evidently young community, which is something that's now a long way from where I am, but very close to my heart. So that's great and something I'm going to try and push as hard as I can and mention during this talk. So I've been involved in antibiotic research for a long time. Penicillin saved my life. I used to be a mycologist and I changed to bacteriology. What I'm hoping to be able to share is some evidence of some progress and some evidence of lack of progress and still something of a broken system, and how perhaps we need to rebuild things from the ground up. And that's going to involve all of us here and more nationally and globally, in my mind. So there have been some positive developments in the UK and the USA. In the US, they are looking to try and pass the Pasteur Act, where the federal government will create a market set of incentives, and antibiotic developers will be paid an agreed amount, yet to be determined, annually from 5 years up to the antimicrobial patent life. And that subscription will be based on clinical need and novelty of the drug. But that's yet to come through. But what has come through already is the UK subscription model. So in December 2020, a year ago now, they announced this Netflix-style model and have selected two compounds, one from Shionogi, cefidoracol, and Pfizer's Cefixia. So that's great. And so that's to give upfront access to the NHS for these antibiotics, and it's not volume linked. So all the right things that one would hope. So that UK model would give something like a £10 million annual subscription from the NHS per antibiotic. So if you actually look collectively across the G20, if those same compounds were selected across the G20, that would give you a $2 to $4 billion market value, so to speak. So the question is that enough? Is that going to be enough? Is it going to be a workable model? Well, as of November this year, Shionogi has no other antibiotics in the pipeline, and Pfizer only has Aztreonam avibactam. So even though those two companies have been selected for this model, it's clearly not enough to incentivize them to reinvigorate a full-on discovery model. So this is problematic. There are other positive lights as well. So Carbex has raised, I think now, over $500 million to invest in antimicrobial discovery. And there are a number of companies here on the left-hand side, Spiro, Entasis, Tetraphase, who were regarded as Carbex graduates who successfully completed Carbex-funded programs to go on and develop effective antimicrobials. The problem is that sometimes discovery success actually equals financial failure. So These set of slides were shown in November 2018 by Kevin Utterson. They're basically slides showing the share value for different companies. And already you can see a black cross next to Tetraphase, because since 2018, they filed for bankruptcy. Melinta has also filed for bankruptcy. Acagen filed for bankruptcy. Navriba is down to a $1 share value. Paratech, when I last looked a month ago, their shares were described on a shareholder's website as a value trap. They looked good value for money, but weren't going to give you anything. And then that leaves you Entasis up at the top here. So let's just look in a bit more detail on Entasis. So this is their share price. It says their share price is currently $2.72. That's last month. I looked this morning, it's now down to $2.03. So what's their track been like? Well, actually, they're doing really well. They've got a sulbactam durabactam clinical trial, phase 3, that went astonishingly well, really low toxicity, really effective against multi-drug resistant Acinetobacter baumannii. So, that is a needed drug. So their share prices started to tick up a little bit until the last quarterly shareholders meeting, which I listened to. They announced that they were actually going to market the drug, and their share price then started to collapse based on that, because that was actually the downfall from some of the previous companies, where they had to pivot away from funding their research to funding sales program. So Entasis are trying to sell this via a new model to try and cut down the huge sales costs that would otherwise be required in a fractured US market. So it's different from the NHS, where you have a single purchaser. In the US, you have a very fractured healthcare market. So you need a big sales force. So that's not terribly encouraging. So let's also think a little bit about what's actually needed globally. We looked at those two subscription models, so to speak, where you had the choice of 2 antibiotics selected basically on UK need. So annually, we've got a mortality of about 52 million from all causes, about 10 million from cancer, around 17 million from infectious diseases, and about 9 of those from young children. So if you actually look at the figures left and right, left are deaths due to cancer, right, deaths due to infectious disease. So we have a burden of infection in countries that perhaps are not best equipped to discover new antimicrobials, and perhaps not be able to pay for new expensive alternatives when cheap traditional antibiotics are still available. It's the access to those cheap ones which has actually broken the model for new ones. So you then go, okay, which antibiotics do you choose if you're going to choose to develop new antimicrobials? So I've just picked a scattergun of antimicrobials, carbapenems in the top, on the left-hand side, cephalosporins in the middle, fluoroquinolones on the right, and 3 different organisms, E. coli, Enterobacter, and Salmonella. So if you look around the world, you get a changing landscape pattern as to where these particular problems are, unless, of course, you look at India, and they have everything in different proportions. So I suppose if you said, let's choose to develop antimicrobials that met the need in India, then perhaps that could tick the box globally. But we don't do that at the moment. The subscription models are based around the specific needs in the countries that have chosen those. So that then becomes potentially problematic, perhaps even within the G20 countries. How do you choose which antibiotics to select for your finance model? And actually, are we missing something in the global south that needs access to effective antibiotics? So if we look a little bit at where antimicrobials are being discovered, these graphs from a patent search for antibiotics from 2001 to 2021 from USA, Europe, and China show an interesting trend. I've squished the axis so they're equivalent across all three. And you can see the only place where there has been a growing increase in antimicrobial patent filing and applications granted is in China. They're decreasing in USA, Europe. The cliff edge in 2021 is somewhat COVID affected, but also just to do with how the data is collected. But it's showing the general trends that actually there's a new kid on the block, so to speak, and China is doing a lot to try and discover new antimicrobials. So perhaps we need a little think about how we and who we work with. So how do we set about trying to be part of the solution? And I think the fact we're all here at the ABX Accelerator meeting is already self-evidential answer to that. We need to work together, and in my mind, we need hardwired discovery platforms that will enable academic and industry groups to work effectively together to push forward early stage discovery. It's too hard, too difficult, too expensive to have lots and lots of platforms replicated within a country and potentially even globally. So we need to think long and hard about how we best use the resources we've got to maximize the number of projects that get pushed through. So this is something based on a program we have between UK and China, really based around the synchrotron at Harwell, Diamond Light Source, and Shanghai Synchrotron, using structure-based informed antimicrobial discovery. So this is fed in at the front end with this lovely atomic force microscopy image from Simon Foster showing Staph aureus exploding, death by physics, rather than just penicillin. Wouldn't happen without the physics. And you have high throughput crystallography. We have a platform where you can take small chemical fragments and screen hundreds of those over a few days. So you have robust crystals of your protein target, and you soak into those crystals individual chemical fragments. And the automated software enables you to identify those fragments very easily. So 500 fragments in a day, that's quite straightforward. That platform can also be used to deconvolute complex natural products. So we set up a system whereby you could take robust crystals, literally dip them into natural product extracts. And if there was a bioactive compound in there that would bind to your target, then you'll find that. So that fragment will soak into your crystal. And so your crystal becomes your chromatography system. That works nicely. We had a simple system here with a beta-lactam soaking into a PBP. And that works beautifully well. And it's a nice way to de-replicate all those natural product hits that we've already found at high throughput. So that can be done screening hundreds and thousands of extracts quite easily, and avoids the need to purify those extracts, which is a great boost. We have set up a whole lot of new biochemical assays, put them into high throughput format down to industry standard. And once we have this in place, we have additional artificial intelligence platform that Frank von Delft has developed at XChem and Harwell. And that will enable a very simple expansion of those small chemical fragments and cheap purchase and then recycling through the screen, through the assays, through the crystallography. So there's a really nice platform there. One thing we would like to add on to that is a technology platform to understand drug permeation, but that's something in the planning still. So that platform in place was actually pivoted by Frank and his team, the wider team, to develop a COVID program during lockdown. When we were all locked down, they were busy working. and they set off from zero in an open source discovery manner to identify chemical fragments to preclinical leads over a period of 20 months. And that involved 30 different groups involved in their fragment to lead discovery program, about 40 groups in lead optimization. And they've now got additional funding to push that forward to the next stage of trials. So that's a lovely example where you have a technology platform that you can use for structure-based drug discovery for antivirals, antibacterials, antifungals, any target, effectively. So for me, that's something that we ought to value and engage with and make the most of as a national resource. Getting towards the end, something else we've started with is instead of doing everything involving IP, I have a spin-out company and we do secret things, but we also do things that are open to the public. And one is this open source antibiotic program we've started now for a year. It's running on vapor. There's no serious funding going into it. And we're pushing forward projects, and my project in particular with Matt, Todd and Frank is the Myoligase project on the left. And we have quite a big team involved now. And we've also had industry buy-in, putting in their stalled antimicrobial discovery projects and their expertise. So that's been really quite exciting. And in my mind, if you have an IP-free open source platform, you can have groups worldwide engaging, which is exactly what happened with Frank and the COVID Moonshot program. And people get recognition by being involved in the GitHub webpage. People can come and go. The information is kept in the repository, so there's no lost knowledge. For me, this looks like a brilliant training platform where you could get groups of PhD students doing their PhDs. One of my students is doing a PhD as part of this, so her thesis is already, being made live as data is discovered. And it's a way to do things to help break down international barriers and maximize resource enabling groups in Africa, for example, where there are no synchrotrons, to engage in structure-based discovery. So almost finally, on the left-hand side is a bit of a bleak post-antibiotic landscape where we're looking and hoping to find antibiotics, and it's not looking terribly attractive. And on the right-hand side, say a Kent lorry park. Instead of having lorries, it would be lovely if these were early stage discovery projects parked, waiting to be picked off and funded through to late stage antibiotics. So that's my final little bit of politics, is that actually I think the funding system is pushed too far down to a few compounds being selected, and that is going to restrict diversity and access globally. And I think it would be great if it was possible to have a funding system where you could push things through to proof of principle in an effective in vivo model, whatever that model is. Not all models are very good. We need better infection models. If you could show effectiveness in a model, that would be great to be able to get to that point and park those and have those funded, and then go back and get another one and get another one. So that's where I'm going to leave that. Thank you very much.